month. And without much ado, I want to introduce the moderator for today's discussion, Dr. Pamela, who is from the Department of Psychology and OBGYN. And if you could give her a round of applause, and hopefully you enjoy the show.
Thanks, Mitch, and thanks to the Graduate Student Association for having me moderate today. It's really a pleasure to be here. And it's nice to see this program continue so successfully. Such a beautiful day, and so many of you are here, and hopefully more than just the Bagels, but you're also here for these excellent talks that we have. Three really unusual studies, and it just highlights some of the great work that's happened at Drexel among the graduate students. So let me introduce the three speakers for today. The first speaker that we have, I'll introduce them all right now, and then between their talks they'll be setting up their different slides and the video presentation that they have, but I'll introduce them all right now. The first speaker is Siddhita Mhatre, and she is going to speak about modeling Alzheimer's disease in fruit flies. Siddhita was brought up in India, and she did her undergraduate degree in pharmaceutical sciences from the University of Mumbai in India. And she came to the U.S. in 2007 for her master's degree in biotechnology from the University of Sciences in Philadelphia. And after she completed her master's, she joined Drexel University to get her PhD in biology. And she's currently in her 4th year of the PhD program in biology. The second speaker will be Maia Smith. And she's going to speak about insularity as an independent risk factor for homicide. Maia went to the undergraduate college of the Atlantic in Bar Harbor, Maine, and lived in a more isolated community on Martha's Vineyard, also in Maine. She's currently an MS student, a Master of Science student in Biostatistics at the School of Public Health at Drexel, and she will complete her degree in June. And she has a special interest in cultural and behavioral change, and has a very interesting talk about this relationship between insularity, isolation, and homicide. Our final speaker will be Edward Damon, and Edward has interests in high-energy physics, and he's going to speak about articulated arm calibration of double Chooz. Edward grew up in Kearney, which is a college town in central Nebraska, and after graduating from high school, he studied physics at Cornell University. And then since then, he's been at Drexel getting his master's and now working towards his PhD. And in his spare time, he runs the Philadelphia Traditional Game Group and volunteers with iPraxis, which is an organization which connects students with scientists and educators. And he lives in Philadelphia, and he wanted to highlight with his cap. But he also has some very interesting work, and he has had the privilege, I guess, with his work to travel between Chicago and also France. And he'll be telling you about that work this afternoon. So again, thank you for the opportunity to be here. Please think of questions you might have for these speakers. After each one speaks, there will be the opportunity for questions and answers, and then some opportunities to address questions to the whole panel at the end of the talk. So right now, let me invite Siddhita to come up and begin her talk. Thank you.
Hello everyone, hope you're enjoying your lunch. Today I'll be talking about aged Alzheimer's disease in Drosophila melanogaster, it's very well known as fruit flies. So why to study Alzheimer's disease? It's the 6th leading cause of death in the US. About 5.2 million people currently are diagnosed with Alzheimer's disease. Apart from that, just the caregiving cost for these 80 patients accounts for $203 billion annually, which is a huge amount for US government. Also, if you see among the 6 leading causes of death, Alzheimer's disease is the only leading cause of death which shows a positive percentage change recorded from year 2000 to 2010, making a real important disease to study. So what's Alzheimer's disease? It's a neurodegenerative disease. It is related to genetics and age. Here, this picture is a patient who was first diagnosed with Alzheimer's disease by Dr. Alzheimer himself. So based on the age, Alzheimer's disease is categorized into 2 different categories. Early onset occurring before age 65 and late onset occurring after age 65. Here you can see a healthy brain and an Alzheimer's disease patient brain. And overall, there is a decrease in brain mass as well as brain volume in Alzheimer's disease patient brain as compared to healthy brain. This neurodegeneration is mainly related to two neuropathological lesions that were first found by Dr. Alzheimer's in his first patient. Those lesions are neurofibrillary tangles, shown up here, which kind of clogs the highway of brain cells or neurons, and the amyloid plaques here, which disrupts the connection between two different neurons or brain cells. So I'll talk more about amyloid plaques today. So amyloid plaques are basically formed through proteolytic cleavage of a bigger transmembrane protein called APP or amyloid precursor protein. Now this APP gets proteolytically chopped by two different pathways, the good pathway and the bad pathway. In the good pathway, it gets sequentially cleaved by two enzymes, alpha secretase followed by gamma secretase cleavage leading to a good peptide. Whereas in the bad pathway, it gets cleaved by beta secretase followed by gamma secretase cleavage leading to this A beta peptide, which is a bad peptide. Now this bad peptide, what it has a tendency to form aggregates, thus leading to plaques. The plaques that are seen in AD patients, which disrupts the communication between brain cells. So as I had mentioned, early onset, so Alzheimer's disease is categorized into two different categories, early onset and late onset. I'll talk about early onset first. So after only 10% of the cases that are reported for Alzheimer's disease belong to early onset group. Much is known about early onset. It is more hereditary and related to genes. Though 90% of the cases reported for Alzheimer's disease are late onset, not much is known about it. The only fact is known is it's well known that it's caused at the later stage of life. That's aging. Aging plays an important role in Alzheimer's disease. So that brings me to the rationale of my study. So based on whatever is known about Alzheimer's disease and adding aging as a main factor here, what we are trying to develop is a model which mimics AD-like symptoms. So in order to develop this model, we are using fruit flies or Drosophila as our model system. So why Drosophila? Even though Drosophila is about 650 million years apart evolutionary from humans, surprisingly it shares 70% autology to human disease-causing genes. It is a well-studied model, it has a very short generation time, it has a wide variety of mutants that are readily available. It is very inexpensive, and one and most important factor is there are many genetic manipulating tools that are readily available for Drosophila studies. One such tool is called US-Galfo system. Now US-Galfo system is a yeast system which has been very nicely adapted in flies. Now what this system is, you have a male fly with a Galfo protein, just imagine as a key, and a female fly with an US upstream or upstream activating sequence, imagine as a lock. And now you have your genes that are placed downstream to your lock. When the fly is made, the progeny would then express your key, that's Galfo, that binds to your lock and opens the door for expression of your human genes. Now, in our system, what we are doing is expressing the bad pathway, expressing human ATP and human base in flies. Flies have endogenous gamma secretase, which chops human APP, leading to A beta plaque formation. Now, what we are using is a very specific promoter, or ELAB, which would express our gene of interest only in the fly nervous system, because Alzheimer's disease is a disease of nervous system. So we are only expressing our genes in nervous system of flies. As I said, US-Galfo is a yeast system which is well adapted in flies. Being a yeast system, it's temperature sensitive. So as you decrease the temperature of flies, growth temperature for flies, you will decrease the expression of the genes that are placed downstream to US. So what we saw was initially when this is how a normal fly looked like. And when we express our genes, that's human APP and human base gene, in nervous system of the flies, at 25 degrees, the flies are born with these defects or phenotypic differences, these crumpled wings and necrotic masses on their mouth as well as abdomen. On quantifying, they show about 60% of these defects at 25 degrees. As soon as I reduce the growth temperature of flies, these defects are reduced significantly, suggesting that we are successfully reducing the expression of human APP and human base genes. Next assay, what we did is something called clamping assay. Now, it is based on the innate response of flies, the innate negative geotaxis response. What it says is, if you have flies in a wire, and if you give them an impact or tap, the innate response is to climb up the wire. Now, what we see is check the ability of flies to climb a predetermined distance in a predetermined time or a given time. What this assay predicts, it just gives an overall view of overall functioning of the nervous system in the flies. It gives the reflex behavior in the flies as well as gives the spatial awareness, because as soon as the flies are put down the wire, they have to orient themselves and then climb. So what we saw here in the blue and the red bars are our controls, and the green bar is our aged 80 flies. As they age, we see past day 40 decrease in their climbing ability. What this suggests is past day 40 there is something that's going wrong in their nervous system. We don't know exactly what is going wrong. So to further investigate whether our 80 flies are actually showing 80-like symptoms, what we did is a coach suppression assay. This is a learning and memory assay for flies. It's a very cool assay. What we do is punish the flies for mating. So we take a virgin male, throw them to a certain time period, and on the day of assay, now this assay is divided into two phases, the learning phase and the memory phase. So in the learning phase, we take the virgin aged fly, male fly, subject it with a mated female. Now the female is mated, so she won't respond to his cue. So the female is mated, she won't respond to his cue. So every time he tries to mate her, she just says no. We do this learning phase for an hour, and we quantify the first 10 minutes and the last 10 minutes of the learning phase. So in the first 10 minutes, as the male doesn't know whether the female is mated or not, he'll try to court her more. And as the hour progresses, if he learns from the behavior that every time he approaches a female, she just says no to him, in the last 10 minutes, he wouldn't try to court her as much. So that's the learning phase. Now, the second phase is the memory phase, where we keep this same virgin male isolated for just 2 minutes for immediate recall memory, and then subject him with the virgin female. Now, if he remembers that every time he approaches a female, she just rejects him, he wouldn't try to mate her, even though the female is receptive in this case. So this phase, we compare with an untrained or sham male who doesn't have a training at first place. So his courtship index or courting, the time he tries to court would be higher as compared to a trained male who has an intact memory. So what we saw in our 80 flies, that as they age, they do have an intact learning. But the problem comes here is the memory. So as they age, they have memory intact until day 60. Past day 60, they have loss of memory, which is again similar to what we see in humans. As they age, they have loss of memory in Alzheimer's disease patients. The next thing what we wanted to check is if our 80 flies have neurodegeneration as that what we see in humans. So we looked at a specific learning and memory center in flies called mushroom body structure. So it's a center for olfactory learning and memory. It is divided into cell bodies, or cranial cells, helix, or dendrites, and lobes, or axons. So I quantified different like surface area of the different structures of this mushroom body structure, which is equivalent to hippocampus of humans. So this is how a normal mushroom body structure looks like. So at day 6, there wasn't any difference between our aged 80 flies or 80 flies compared to a normal flies. As they age, if you see, there is a weakening of lobes or axons, and that can be quantified with a significant difference here. And as they age to day 100, these axons are missing, which can be then quantified here, and even I see a decrease in dendritic field. So to summarize, we overall could recapitulate all the symptoms that are seen in humans in flies now. We see spatial awareness, which is equivalent to decrease in climbing ability of flies. We also see age-dependent neurodegeneration and an age-dependent loss of memory in flies. So what's next? We would like to see whether these flies have amyloid plaques that are formed in humans, that are similar to humans. And the use of this model, what we plan to do is for rapid in vivo testing for pharmacological testing, like small molecule drug screen, and to test putative genes that are associated with Alzheimer's disease. So I would like to acknowledge my mentor, Daniel Miranda, different company members who have helped me with my project, my lab members, and different undergrads who have worked with me. And I would like to take questions. Does anyone have any questions for our first speaker?